Have you ever looked in the mirror and thought, I have my mom's eyes, or I definitely got my dad's smile? Well, the reason why is all thanks to a hidden code, a set of rules for life itself. And what's amazing is that this code wasn't cracked in some futuristic lab, but in a quiet monastery garden over 150 years ago. Today, we're going to crack that code together. So, here's the game plan. We'll start with the man who figured it all out. Then, we'll learn the essential lingo so we're all on the same page. After that, we'll dive into his big experiments, understand the laws he came up with, and then it's your turn to put it all to the test. Okay, let's set the scene. Our story starts with a guy named Gregor Mendel. And he wasn't some world-famous scientist in a fancy lab. He was a monk living in a monastery. But he had this incredibly curious mind and a real passion for science and nature. And this timeline, wow, it tells such a powerful story. Mendel starts his game-changing work with pea plants way back in 1857. He publishes his findings in 1866, and nothing. Crickets. The world just wasn't ready for it. His paper was basically ignored for over 30 years, and he passed away long before anyone realized he'd laid the foundation for all of modern genetics. It just goes to show you, sometimes genius is just way ahead of its time. All right, to really get a grip on Mendel's genius, we first need to learn the language he basically invented. So let's run through a few key terms. Think of these as the building blocks for everything we're about to cover. First up, the allele. It sounds complicated, but it's not. Just think of it like this. If a gene is a category, like eye color, then the alleles are the specific options in that category, like blue or brown. For Mendel's pea plants, the gene for height had two alleles, one for tall and one for dwarf. We usually show them with letters, a capital for the dominant one and a lowercase for the recessive one. So what's this dominant and recessive stuff all about? Well, a dominant allele is kind of the boss. If it's there, it's the one you see. So a plant with two tall alleles, or even just one, is going to be tall. A recessive allele, on the other hand, is a little shy. It only gets to show itself if it's paired up with another recessive allele. It gets masked by the dominant one otherwise. And that brings us to two absolutely crucial terms, genotype and phenotype. The genotype is the genetic code itself, the actual letters, like TT or TT. The phenotype is the physical thing you can actually see, the tall plant. So an easy way to remember it is the genotype is the blueprint and the phenotype is the actual building. Last bit of vocab, I promise. How do we describe these pairs? Well, if an organism has two of the same allele, like two talls or two dwarfs, we call it homozygous. Homo just means same. And if it has two different alleles, like one of each, we call it heterozygous. Hetero means different. See? Simple as that. Okay, now that we've got the lingo down, let's see how Mendel put it all into action. We're going to start with his simplest, but maybe most brilliant experiment, the monohybrid cross. That's just a fancy way of saying he was tracking one single trait at a time. So here's what he did. He took a purebred tall plant, meaning it only had tall alleles, and crossed it with a purebred dwarf plant. Now, you'd probably think the baby plants would be somewhere in the middle, right? Some medium height? Nope. What he found was that every single one of the offspring was tall. Every single one. That was his first big clue that the tall allele was dominant. But then came the real genius move. He let these new tall plants self-pollinate. To figure out what would happen, we can use this handy little tool called a Punnett square. It's basically a prediction grid. You put the alleles from one parent on top, the alleles from the other on the side, and then you just fill in the boxes to see all the possible combinations for the next generation. And check this out. This is exactly what Mendel saw in his garden. When he counted his plants, he found a super consistent pattern. For every one dwarf plant, there were 3 tall plants. That dwarf trait, which had totally disappeared in the first generation, was back. This clean 3-to-1 ratio wasn't an accident, it was a massive discovery. But what was going on genetically under the hood? Let's look at our Punnett square again. You can see there's one TT combination, that's a tall plant. There are 2 TT combinations, those are also tall plants. And then there's one TT combination, our dwarf plant. So that 3-to-1 ratio we can see is actually caused by a hidden 1 to 2-to-1 genetic ratio. It's a great reminder that looks can be deceiving. You think Mendel stopped there? No way. He got more ambitious. He started wondering what happens if you track 2 traits at the same time. Like, does being a tall plant make it more likely that it will have smooth seeds?
Or are those things totally unrelated? This question led to what we call the dihybrid cross.
For this one, he started with a plant that was purebred for two dominant traits, tall and smooth seeds, and crossed it with a plant that was purebred for two recessive traits, dwarf and wrinkled seeds. And just like before, that first generation of offspring only showed the dominant traits. They were all tall and had smooth seeds. Then he did the same thing again. He let this new generation self-pollinate. Now our Punnett square has to get bigger. It's a 4x4 grid with 16 boxes. That's because each parent can now make 4 different combinations of alleles to pass on. This grid lets us map out every single possible genetic outcome.
So after counting literally thousands of pea plants, Mendel found another beautiful, consistent pattern. Out of every 16 plants, he'd get about 9 that were tall and smooth, 3 that were tall and wrinkled, 3 that were dwarf and smooth, and just 1 that was dwarf and wrinkled. This elegant 9 to 3 to 3 to 1 ratio was the proof he needed. It showed that the gene for height and the gene for seed texture are inherited completely independently of one another.
So, from all this incredibly careful work, from all this counting of peas, Mendel was able to figure out 2 fundamental laws of heredity, the rules that govern how traits get passed down. These laws are basically the pillars of classical genetics. The first one is the law of segregation. So what does that actually mean? It's simple, really. It just means that for any trait, the 2 alleles a parent has get separated, or segregated, when they make their reproductive cells. That way, their offspring gets 1 allele from each parent, a 50-50 shot at either one. And his second big rule is the law of independent assortment. This is exactly what we saw with the tall and smooth peas. It means that genes for different traits get sorted independently. The allele a plant gets for its height has absolutely no effect on the allele it gets for its seed color. It's like flipping 2 different coins at the same time. All right, you've heard the story, you've learned the vocab, and you know the laws. Now it is your turn to be the scientist. Let's see if you can put all this awesome knowledge into practice. Okay, here's your first challenge. Imagine we cross a purebred red flower with a white flower. We know that the red color is dominant. So what are the baby flowers going to look like? What's their phenotype? Go ahead, pause for a second, maybe sketch out a little Punnett square, and see if you can crack it. So did you get it? Let's walk through it. The red parent can only pass on a big R allele. The white parent can only pass on a little R allele. That means every single one of their offspring has to have the genotype RR. And since we know red is dominant, the phenotype for all of them will be red. They'll all be red flowers. So we've traveled all the way from a humble monastery garden to the fundamental laws that govern life itself. It all started with just patiently counting peas. And that leaves us with a pretty mind-blowing question, right? If these simple rules let us predict traits in plants, what does that mean for our ability to understand, to predict, and maybe, someday, even to influence the incredibly complex traits in ourselves? The code has been cracked. What we do with it, well, that's the story of our future.